First of all, I know many of you are already aware, but I'm just trying to recapture them to refresh your minds if somebody, at least one person, is still studying all these details. So when you say proteomics, what is proteomics? Is basically the study of proteins, or the study of the protein flux in the cells, and which is called the proteome. And when you say mass spectrometry, according to John Fenn, how he defined mass spectrometry is the art of measuring atoms and molecules to determine their molecular weight. And what is this art John Fenn was talking about? So he basically was trying to elaborate that during a mass spectrometry experiment, uh, uh, atoms and molecules are ionized, and they are separated based on the M over Z value and detected with respect to the M over Z values. There are many ways that you can ionize atoms and molecules. There are many ways that you can separate ions based on the M over Z value, and there are many ways you can detect them. So depending on the application, you can ionize molecules in different ways. For instance, either electrospray ionization or matrix laser desorption ionization, or chemical ionization or electron ionization. And also there are many methods that we can basically separate ions based on the M over Z value, where you can separate them when they are isolated in a magnetic sector, or you can channel them through ion quadrupoles and how they distribute themselves in various electric fields, and how you detect the ions, whether it's a direct photoelectric effect based methodology, or is it an offline resistant change that you detect to see the oscillation changes of the ions, so that you can do a deconvolution to get the M over Z ions. So at the end, what we get is a mass spectrum. So when you say proteomics mass spectrometry, then it's basically how we use mass spectrometry to profile proteins or study proteins. After compiling all these terminologies together, in proteomics mass spectrometry, we basically ionize proteins, we separate them based on the M over Z to basically eventually generate a protein mass spectrum. Then we can say quantitative proteomics mass spectrometry is the way how we use mass spectrometry for quantifying proteins. We look at the general direct protein analyzing mass spectrometry platform, where you can use a protein solution to be mobilized and to ionize using, for instance, here electrospray ionization, and separate and detect the ion in the mass spectrometer to generate a typical single protein mass spectrum. So these mass spectra can be deconvoluted to identify its molecular weight, and if there is a modification, we can identify that as well. So using this kind of a workflow, we can get an idea about the level of protein modification, and we can have a good understanding about the relative quantification of if there is a modification of the protein, and what is the extent of modification. However, multiple modifications can complicate these spectra, as you can imagine, and data interpretation becomes more convoluted. So and also we have to consider only well ionizing proteins can be studied, and for instance, membrane proteins typically do not provide good mass spectra. So for membrane protein applications, it's a bit challenging. In a general bottom-up proteomic approach, what we typically do is we make a we do a proteolytic digestion of a protein solution. We separate the peptides in a chromatography column. As the peptides come out of the column, we basically ionize, and we do something called data-dependent mass spectrometry. So in data-dependent mass spectrometry, mass spectrometers has evolved significantly to do so many different experiments. Typically, what it does is it generates a survey mass profile to have peptide ions, and we can let the mass spectrometer to pick the most intense ions and do selected ion fragmentations in different time segments. And we can repeat the entire cycle until all the peptides are eluted from a column. Then to identify these or to assign peptide sequences for each of these experimental MSMS profiles, what we typically do, we select a database of protein sequences, generate theoretical mass spectra, and we match them with the fragmentation data to come up with the list of protein IDs, peptide IDs, their probabilities and quantities. Now, if you look at this entire workflow, there are two main places where we can use a response of the peptides to quantify how much peptide is a particular peptide is there. One is the survey MS profile. The other one is the fragmentation profiles. So what I want to really highlight here is peptides can be quantified using responses of the survey MS profile or responses obtained during related fragmentation MS signal. So protein quantification can then be derived from peptide responses, basically. And once you do this kind of a exploratory experiment, there's another method called targeted mass spectrometry, where you repeat the exact same experiment, where you know that some peptides can be identified using the technology, and we can target those ions to monitor only a single ion or its fragmentation ion. So in the single ion monitoring approach, which is abbreviated as SIM, you can generate an extracted ion profile. Again, doing repeated fragmentation of those ions, you can utilize one fragment ion or multiple fragment ions to do single reaction monitoring or multiple reaction monitoring experiment, again, to do an extracted ion profile of a particular solution. So the SIM is more sensitive, of course, because it is not fragmented, because during fragmentation, you lose some of the signal. However, co-eluting peptides with the same mass can interfere when they are eluted during the chromatography. But interferences can be minimized if higher resolution settings are implemented. So it depends on the mass spectrometer settings. And SRM, MRM, less sensitive, of course, compared to SIM, but very specific due to the use of peptide-specific fragmentation signals.
Okay. But I would like to mention here, so you did a really nice job walking through that. But if everybody just takes a minute to notice that the y-axis on all of these graphs, when you do mass spectrometry and you're focusing on measuring the M over Z's, that you get an intensity value. So the intensity value is typically representing like the total number of ions. And so then that's really where we get this information. So this is something that is inherently different than quantitation by high throughput sequencing. And so I want to take a moment to just mention that, because we spent a lot of time talking about quantitation by high throughput sequencing. So quantitation by high throughput sequencing uses read counts. So you just count the number of times that you see a molecule. And so what's interesting about mass spectrometry is you can also use a counting method. So I imagine that Dr. Wisharatna might get to that. But in addition to that, we actually can get a measurement of the total intensity of the number of ions, right? And so that's an inherently quantitative value, and it's very different than what you get with high throughput sequencing.